Okay, so it's 10 past 9. We are at a water depth of 4,910 meters right now, 60 meters off the seafloor. On our navigation screens, what we can see is that we're about 80 meters northeast of our primary target for the start of the day, the Hot Chimlets low temperature vent site. So what we're going to do is to transit for the first 50 meters of that to the southwest to go over a ridge that we can see on our bathymetric chart, which is shallower than the vent site we want. Once we're over that lump, which you can now see on your video screen, you can see Nereus, the little white icon, is going to be going southwest over that little orange blob. It's a topographic high. When we get to the yellow contour the first time, that's the 4,980 meter contour. We're going to drop down to the seafloor and then drive the last 30 meters across a little gully, and then we should be arriving on the north face of the BB Vents hydrothermal mound. And what we're going to be studying today are the relatively low temperature diffuse flow sites, which can still reach temperatures at over 100 degrees Celsius. So that's hot enough to boil water at atmospheric pressure on the Earth's surface, but at the depths we're at, too deep for the water to boil. Just to give you an idea of what's in store for today's dive, our first objective is to conduct some low temperature venting sampling. So we know that the whole of the hillside on the north face of the mound that hosts the black smokers we were sampling yesterday is pretty much alive with diffuse flow and teeming with biological life. So that's the sites we're headed to. The work that our grad student Jill McDermott's been working on with Jeff Seaboard in particular at HROV has revealed all kinds of interesting complex organic molecules present in these hydrothermal fluids, even though they're extremely hot. So we're interested in both the organic geochemistry of those samples, so we want to collect more of them, so we can study them in more detail. And then in parallel with that, Julie Huber and her colleague Julie Revaillot, who's out here at sea with us, are interested in what kind of microbial life takes advantage of the unusual chemical conditions we see there. So they're going to be conducting microbiological sampling using a new version of CHIP supersampler. Yesterday we used CHIP supersampler for geochemical studies of dissolved particulate interactions in the young black smoky sections of hydrothermal plumes. Today we have the supersampler reconfigured in a different mode, and that's for doing microbiological studies. So we're going to be doing that for about the next 3 hours. We only have a very short dive today, I'm afraid. It's only 5 hours in total. So we have 3 hours available for geochemical and microbiological sampling, and then we're going off for a bit of exploration work that when we were here in 2009, we had an unplanned event where we left one of Jeff's titanium fluid samplers on the seafloor about 500 meters west of the now known vent site. So we're going to be heading over there to see whether we can't locate that and recover it and bring it back. So that's the plan. 3 hours of sampling. We should have some very photogenic vent sites coming up very soon, and then we're going to go and spend the second half of the cruise, or the second half of the dive, seeing if we can't find Jeff's sampler. So we've got a rather stripped down basket today. We have the super sampler, we have 2 of our fluid samplers, and that's it. And then hopefully we'll actually have more samplers to go home with this cruise than we came out with, which will be the inverse of the last time we were here with Nereus 4 years ago. Okay, we've completed our 50-meter move through the water column. It's just approaching 9:20 here aboard ship. We are at 4,910 meters, 70 meters above the seafloor, and we are heading down to the seabed at a depth of about 4,980 meters, which is about the target depth we were expecting to find our hot chimlet site at. So we should have the seabed in sight in the next 10 to 15 minutes. So it's 9:24. We're 10 meters off the bottom, and the seafloor is coming up. We have a world of. I have a nice little patch of shrimp. Is that, or is that mineralogy on the left? Oh no, that's just, yeah. There's a little patch of shrimp just there on the left. So we've arrived on the seafloor. We're in the area of Hot Chimlet, very rugged terrain. but some areas of diffuse flow. That's it straight ahead. That's where we want to be. Actually, we're already there.
Well, for us. I'll be right back.
Bottom is soft as well. I think 20 or
So
30
we've
minutes after we got the water.
yeah he. So, Jeff, I'm not sure if this is exactly the hot chimlet site. If you want to come and have a look, but it looks pretty similar. We have a big cluster of shrimp just here. I think there's some low temperature flow over to our left as well. If you can spin left, Casey. In fact, there's our marker just to the left as well, or it's somebody's marker. We definitely left a marker at the hot chimlet site. Oh no, that's one of the ISIS markers from February, I think. We'll let the dust settle. Oh yeah, and I can see our diamond-shaped marker over to the left as well, just left of the manipulator.
Doppler or the last of it? We're going to get rid of a weight on the Doppler.
Okay.
So that we end up crossing
Yep, so we're just adjusting our... So we have two markers in the same location, so we've seen both our own marker from when we were here in 2012, and it looks like a second marker has been dropped right next to it. Perhaps measuring some long-term experiment that's in place. From the handwriting, it looks very similar to the one we saw yesterday from the UK cruise that was here in February of this year. So we're just letting things settle out here, but this is a very good start to the day. I was saying earlier we have to be very efficient. We only have 2.5 hours for our sampling area, so it's kind of neat that we've landed right where we wanted to be. So kudos to the team who navigated us into the right place. We spent a lot of time last night going over our navigation offsets, and apparently the voodoo worked.
That's all it is.
So there's a lot of voodoo and a little bit of applied mathematics that goes with it. So you know what we're doing. Casey is starting his basket origami early this morning. So we've taken some extra ballast down today to counterbalance the weight of the fluid sample we want to pick up for Jeff Seaward later in the day. So we're just adjusting that so that we will not be too heavy now, so we can actually hover close to above the seafloor. Okay, so the time is 9:32. We are about to get ourselves settled up. You can see a lot of fine-grained metalliferous sediments being stood up in the water column just now. We are gonna wait for things to clear, and then we can get started with our sampling here at the hot chimlet site. So things are beginning to clear slowly. Say what part, Casey?
What you said you saw a marker on?
It was on the right hand side of your view just now.
Right hand side of that.
Yep. I think it might be a little bit uphill to the right. You see there's some shrimp flying around up there. There's one marker there, bottom right there. Some fluid flow right at the base of where that is. You see the white patch will be shrimps. That'll be where the diffuse flow is that we want to sample. There's some lumps of rock between us and there. Once we were flying in, the first thing we saw was a patch of white shrimp.
Yeah, I think we're left of that.
So that's really what we want to go to, is where the shrimp are in the diffuse flow. These are basically, this whalebone experiment's been put out to see if any animals colonize that at some point in the future. Is there a way, could we actually just try while we're there, sorry, to looking at those bones, if we could try the zoomed in camera briefly, just to collect some data. One of the things they want to do is to see whether any Osedax worms colonize these whales. So since we're here, we could actually share this data with Diva. We know Diva Amon was part of a cruise that I was the science lead for about 3 years ago for the Natural History. She's at the Natural History Museum in London. So what we're going to do now is switch to our zoom camera. We have two different cameras that were donated by James Cameron recently. So if we can we can still pan, right? Can we pan right as well? And then if we can focus that. Yep, so that's hydrothermal shrimp around this area. So these are deliberately placed whale bones. We know from past experience that the way animals move from, or it's hypothesized that the way that animals move from a vent site to a seep site and colonize their way around the planet, they take advantage of other things, and one of those is whale carcasses, that when a whale dies and drops to the seafloor, the rotting of the whale can actually sustain ecosystems of some of the same species that you find at vents and seeps for up to 100 years at a time. So these are historic whale bones that have been placed out to sea adjacent to this hydrothermal area here called the Hot Chimlet site to see what's growing. These were only put out in February, so this is just useful time series. We're just collecting some video now and recording it that we can share with Diva when we get back, to show that some shrimp have shown up, but apart from that, how long have they been there? About 4 months or so. Currently nothing much else going on. So that's it. Thank you, Casey. We just want to get a look at that, and we can switch back to our zoomed out camera and get on with our own sampling for the day. But since we were here, that's a little bit of serendipitous fieldwork we were able to do for our UK colleagues. The visual cues to follow, Casey, are wherever you see little white patches on the ground where the shrimp are clustered, that's where the types of fluid are that we want to go to today. And there are no bad patches to study as far as Jeff and Julie are concerned for today's dive.
covered in metalliferous sediments. Metalliferous. I think he probably just went to get some coffee. Adrian Glover is asking if we can get a still of the bones. I think we did, right?
We got about a minute of high
Okay.
definition video with
I will let.
camera, and we can do frame grabs from this.
Okay. Adrian and we got some high definition video. We can see marker 23 in sight. That's a Jason marker. Oh, sorry. It's because my head is smaller than everybody else's. Is that better? That's better, right? Okay. So we are in the hot chimlet area, looking for the point of venting. The marker you see in front of you is something we put on the seafloor in January 2012. It's basically a nylon rope with a little weight at the end to help us find our way back when we come back or other scientists come here. We share our navigation maps with other groups. We're just going to sit down and wait for things to clear so that we can see what we're looking at. How did we come up with the name Hot Chimlet? Was it because it was the way it looked? I came up with it? No, it's not the answer I remember. Apparently I named this vent last time we were here. But my recollection was I had the mid to 4 watch, and it was 3 a.m. is a good time to name vents. I do like the word chimlet. So we are at 4985 meters. We're about 30 meters deeper than yesterday. We're still in the BB vents area, right? There's BB vents, BBC, and BB Woods, all named after the naturalist William Beebe. Right now we are just waiting for the flurry of sediments that we stirred up to settle down. We're looking at a marker, although you can't see the marker in your camera. There we go. There is? Yep. All right, we see some shimmery water off to the right of the basket. Last year, the max temp we got here was 150 degrees Celsius. So we're going to try to get a couple of water samples here for Jeff Seawald and for me. So things are beginning to clear down here. We can see some shimmery flow at the base of the marker. Shimmery flow means we have hydrothermal venting, which is what we're interested in. I don't know. Oh, that's right. Doesn't look like it. Is it possible we're right on top of it? Yes. So we're just trying to figure out the best approach for sampling this. Hey, this is pretty helpful. Yeah, we're like right on top of it. This is what Adrian just sent me. So we're probably a meter or two on top of it. Adrian was here in February and he just sent us a picture of what he recalls this site looking like to help us find our bearing. No, I want to go second. Just because I want to let Chip sleep longer. Yep, shrimp on the seabed. No, that's fine. So, looks like the most vigorous flow is right in front of the basket, so we're just backing up a little bit. We're gonna then get out our temperature wand and check it out. Metalliferous sediments. We can also call them hydrothermal sediments. You know, it's fallout. Okay. These look metalliferous though, don't they? They look rusty. All right, so you can see us sort of settling back over this area. It's not clear to me why I named this chimlet, since I don't see any chimlets, but...
Save that story for when you see Old Man Tree later in the week.
Yeah, right.
I think there was a little lip
Yeah.
and it had a tiny little cute little chimney.
It must have. It must have.
It was very nicely named, and I had naming envy. That's how I know it knew the name. I took over the watch on the 4 to 8.
So we're going to settle down here and get out our temperature probe, sniff around a little bit. We're pretty sure last year we got a max temp of about 150 degrees. So we'll see what we can get here this year. Oh, no? Just getting a little bit organized here in the control room. It's actually a light crowd this morning. Many of us were up quite late, processing samples and turning the vehicle around. I went to bed at 2:30 and Cindy and Max were still up. Chris and Julie were still up. Yeah. So we are at 4985 meters. These are the deepest vents at this site, right? This is an area called Hot Chimlet that we found in January of 2012. And we're going to poke around and take some temperatures. The view you have is courtesy of James Cameron's cameras that went to the bottom of the Mariana Trench on his Deep Sea Challenger sub. He donated a submarine and all its goods to Hui, Woods Hole Oceanographic, and we're using them on the deep diving Nereus vehicle. Now you can see the hot fluid coming out of the seafloor, all the shrimp around it, some white staining on the rock. That's probably a mineral precipitate of some sort, right? Yep. You can see the shrimp are just around the periphery, probably where around 25 or 20 degrees C, I believe, is their maximum temperature. All right, we're just going to take some high-def video for a few minutes here. The Hot Chimlet site, 4985. This is the deepest we brought the vehicle so far on this cruise. Nah. You think it's hotter than 150? Well, I think we've determined that it's cooked shrimp. Isn't that what we figured out last time? It's too hot for microbes. Yeah, I think you can get a good sample. Are you going to put both your samplers here, Jeff, or see how it goes? Okay. Yep. We're going to use an IGT here, measure the temperature, and take a sampler. Well, Hot Chimlet was named hot for a reason. It's not very big, but it looks hot. We're taking bets on how hot it is. I'm saying 150, which is a very educated guess, given how hot it was last year.
I'm going to go with 186. You're a ringer.
Layton's going for 186. We got 116 from Casey, the pilot.
I'm going over 200.
And Chris German is going over 200. Pardon? Jeff Seawald's 153. And Sean, 155. All right, we got our bets.
Okay, the winner gets a can of alcohol-free O'Doul's.
Yeah.
150. And Sean was 155.
You got previous data though, haven't you guys?
We do.
They are ringers. They're hustling us. They're event hustlers. Because the game was given away by the fact that we already had a market here. Okay, let's zoom back out. Let's switch cameras. Thank you very much.
All right, we're going to get an IGT out of the basket and take a temperature. We call it sniffing around the vent with a snorkel. Has a temperature sensor. using something called an ICL, a inductively coupled loop. It's actually pretty cool. It's a magnet. There's a little loop sitting on the basket of Nereus that talks electronically to the sensor at the end of the sampler.
No, we're going straight for an IGT, yeah, they changed their mind. Or both. Just trying to be efficient so he can actually go get his other bottle later.
He has ulterior motives.
We're breaking up at midday for the installation arm.
What time?
Midday. We have to go and do that.
The
The captain says we have to come up at 2 o'clock. It's just diffused all the way.
Okay. So we have a question from Twitter asking any idea what the length of those shrimp are. And I do have an idea because I watched Cindy Vandover dissect about 150 of them last night.
Those was
And they were
tiny ones.
tiny. They were an inch, maybe, an inch long. We've seen bigger ones, I'd say up to 2 inches. She was surprised at how small they were, and they were a lot of work to dissect.
The thing that got me was, did you see those acid fish?
Yeah,
The one that Cindy was going to put her finger in its teeth?
yeah.
Did
Ugh.
you do that?
I didn't do it. I'm scared of fish.
But the finger only just went in the... It only just fit... I mean, I thought they were like huge things because they devour all the shrimp and then you see it in real life. It's like, wow. I could only just get my...
No, the fish was about 8 inches long, I'd say. But still really small head and...
How do they get the shrimp? They eat them in bits, or
I guess so.
are they one of those animals?
They
that
chew
eat
42 times before they swallow. They do what their mummy told them.
Is it a specific part of the shrimp they eat, like some on the soft
Shrimp ears. It's a delicacy. It's a delicacy at some bedsides.
Actually, the size of the shrimp is really important for the biologists because they're using that to figure out recruitment and reproductive strategy for different vents in different areas. And one of the things that Cindy and her British colleagues have been studying is what's the relationship between the shrimp at this site at 5,000 meters versus the nearby site at 2,300 meters, Van Dam. What's the size distribution? What can that tell us about their reproductive strategy and things like that? So when Cindy's up later, maybe she can talk about that because I should probably not say much more
But I know
because I'll be
she-
wrong.
She had lots of ideas about midnight last night.
Yes.
What other samples she might take of what she should now do now. Based
Of
on the
small
science
shrimp.
evidence,
All right, this is IGT number 4.
Still may have a quick computer swap, so.
We have a follower on Twitter who's guessing that it's going to be 250 degrees Celsius.
Okay.
Hotter than me.
Someone on Twitter who guesses what?
250. Stanton Belford.
Have they been here before? They're not part of the UK team that put out the more
They're not on
recent.
the Shinkai, they didn't.
Nope, Stanton followed us yesterday and is following us again today.
Hi, Stan.
Hi, Stan.
Oh, you're getting a bunch of shout-outs.
Keep those questions coming.
Is today Julian Day 176?
Today's Julian Day is 176.
I know what day it is. What time, maybe not.
It gets crazy when you start referring to days to Julian Day. Days that are mean, you know, tell people have no meaning as they.
Julian Day. I can barely keep track of time when I'm out here. I woke up and looked at the event log to try to figure out if I should get out of bed, and I had a lot of trouble translating the time, you know, to what was on my phone.
Yeah, I should really do. I should do a brackets. No, they have to figure out a local time.
See, you should have gone to YouTube. You could have heard my announcements.
Oh.
Stream the video.
Well, I didn't want to wake
Happening
Cindy up.
further down the
You
corridor.
can actually stream the video on the ship.
I got an email saying that it's not going in. So it went off on my phone, looked at it, and went, thanks, Chris. It's so good.
Are you guys ready? Yeah.
All right, we got our temperature
Going in.
sensor in the vent. We're going to use it to...
Okay, hold on. We're fluffing.
Just kidding.
What'd you say, Sean?
Starting the text logging file.
Oh, okay.
Can you feel me?
All right. So Nereus is holding in its arm a isobaric gas tight sampler from Jeff Seawald's lab. Allows for the collection of fluids, keeps them at the temperature they were, keeps them at the pressure they were collected at even when we bring them back up on the ship, and that allows them to get accurate concentrations of all the dissolved gases. Last night they were very happy with their 398 degree black smoker fluid. Now we're looking for something a little bit cooler. Jeff's in the back of the room shouting temperatures. We're up to 50 degrees so far.
Basically, you have to put it in. Nothing can't ring your own back.
Stop before you get to 200.
121.
121 so far, which is the upper temperature limit for life. Microbial life, that is. Our colleague Ken Takai, who's diving here, dove here a couple days ago, cultured a methanogen that grows at 122 degrees Celsius when put under pressure.
And we're just going to cycle the cameras so we can see the artist.
Okay, we're just going to cycle to our zoom-in camera. Highest we've seen so far is 121.
That's when I get the big bump.
Turn off the port outer, turn on the starboard, full starboard side lights. Turn on the port outer. Port brown.
You see the little black lines? That's the crack.
What's the temperature now? 70.
So far, the highest we've seen is 121. Yeah. Can't let the pilot win. So far, right now we're in the mid-80s. It's really hard to tell exactly where the intake is because of that shimmery flow. It sort of messes with your vision. So sort of always trying to find the sweet spot.
The right was where I got 120.
Oh. What's going on? Ooh. This time it looks like we've sort of stuck it in the crack a little bit. Gonna give it a shake so we don't have any sediment left on it. Well, it's hard to tell if the shrimp are really in it, right? Because, I mean, it's this really, really steep gradient. So, you know, you have this hot fluid coming out, and you probably just have these tiny little seawater circulation cells. So sometimes it looks like they're passing through, you know, 150-degree fluid, but they're not, right? Yeah, they're right around the periphery. Yeah, you can definitely see when they're getting burned. They're very, yeah, they've got a lot of sensitive receptors on their surface. So we're still having a little bit of trouble with our finding the sweet spot in this vent, but we're still working on it. Patience is a virtue in deep sea oceanography. If you think about it, we're controlling a vehicle that's 5,000 meters beneath us. That's mainly connected to us by a fiber optic thread the size of a fishing line. So it's okay to be patient. You must be patient. All right, we're at 75 degrees Celsius. We know it's hotter, so we'll keep looking. We got to make sure Casey gets a good workout this morning on the arm. 87. I think just up into the Did you think just up the crack a little bit? Doesn't it look hotter? It's more focused. Ooh. We're just scooting rocks around the seafloor to make the best venting orifice for our sampling needs.
Dark grey is probably the thing to aim for because they can't be light, but it's going to be fresh sulfide mineral
Yep.
rather than neon.
Yep. That's been our experience.
It's a question. As you build out the quantum you say it's shrimp. Is it something that, white stuff is right in there low?
Well, not being a mineralogist, it would not be worth it for me to pick up that rock, but maybe Max or Meg Tivey, our colleague back in Woods Hole, who's doing some of the mineralogy down here, would be interested. We certainly got a few of them last year. And there are other scientists working on that as well. But if it's a pretty, yeah, it's nice to have paired fluid samples and rocks, or when you have a chance to do that. Yesterday we got some sulfide from the high temperature chimney we sampled. So we're having trouble with the temperature sensor on Jeff's sampler. He and Sean are trying to work it out in the back of the room. Those of you just tuning in, we're at the world's deepest hydrothermal vents. We're at 4985 meters. We are located in the Mid-Cayman Rise, the Caribbean Trough, just south of Grand Cayman Island in the Caribbean Sea. It's about 100 degrees outside. It's really hot here. 100% humidity, sunny, nice flat calm day outside. But beneath the seafloor, it's dark. Ambient seawater is about 4 degrees Celsius. But we find oases and hot springs of life at these deep sea hydrothermal vents. And right now we're trying to sample one called Hot Chimlet. We just discovered it in 2012. Put a marker down here. We're back to get some more samples from it because it has some very interesting fluid chemistry. These vents are extremely high in hydrogen, which is hydrogen's played a really important role during the origin and evolution of our planet and life on our planet. There's lots of microbes that can use hydrogen to grow. One example is methanogens. They're part of the archaea domain of life. Archaea look a lot like bacteria. They're tiny, small, single-celled organisms. They divide by binary fission. They don't have any membrane-bound organelles. But actually, archaea and bacteria are as different from one another as we are from them. Archaea have different cell machinery. They make their proteins differently. They have really unusual lipid membranes. And all the methanogens belong to the archaea. And they can use hydrogen and CO2 to make methane. And that's sort of all they need to live. Oxygen is not a nice thing for them. They're obligate anaerobes. And our data from this site in previous years indicates there's a lot of methanogens in these fluids, which makes sense since there's a ton of hydrogen available. And there's also a lot of carbon dioxide. What'd you say? So we're swapping out some instrumentation on Jeff's computer to see if we can get better comms with our temperature probes.
Yep.
Okay.
That's good point.
Could you tell me what time it was when we did the zoom in video, and also what time it was when we did the
So we're going to wait for Jeff to do that.
It's a nice place, this
Hot
Hot Chimlet.
Chimlet is a nice place.
Nice place to hang out.
If you're going to be a seafloor tourist, it's a good spot.
Nice view out over the valley in the north here.
Right.
It's not south facing.
Right, we started at 1354.
Which one?
This was the last one we did.
Okay.
And then we ended at 1357.
Great. And the wellboat?
Around 1336. We zoomed in at 1336, 57. And we finished at 1338 and 24.
Yeah, pretty picture I took last night.
That was beautiful. I just saw it on Twitter.
Oh. That's right, I tweeted it.
It's amazing. We sat next to each other. That picture. I saw it a few moments ago on Twitter.
Yeah, well, Twitter's interesting, right? So we are paused here for a minute while we figure out what's going on with our temperature probe. We're having a little bit of trouble communicating with the ICLs on the vehicle and on the sampler. John Howland's in the back of the room helping Jeff diagnose the potential problem.
I'll have that live action shaky feel.
All right, so we're still in the back of the room trying to figure out what's going on with our temperature probes.
So this is a place called Hot Chimlet. It's a slightly lower temperature than but they're still over 100 degrees Celsius. So hotter than our boiling. And what's really cool about them is it's the same fluid, basically the same cold fluid. It's not at the black side of the sample is the but what this stuff actually has in them is somehow they as they cool down the ingredients of it. Some of the things like the methane, it's very high methane in all the samples of hydrogen. At intermediate temperatures, they actually react and start generating the complex organic bonds. So this is the stuff. This is the stuff that's specifically linked to life and the work we're doing. We're trying to understand whether we built a system like this that like originated out spontaneously just with rocks and water and start making more building blocks for life. So these are the samples. We had to collect the end of the sample to understand what the mixing is. But the stuff we did yesterday looked really cool and looks scientifically. This is the much more interesting side. So this is really we have today and 2 more dives. We'll come back here later. So the next 3 dives are all about sampling these kinds of sites. So it's kind of cool. It's like about 120 degrees.
So far the highest we've gotten is 121,
So
but our notes indicate it should be higher.
what? Geochemistry reactions of life, and then everything from about 120 degrees and cooler or 2 degrees, which is the hottest that life can exist. So she's the microbiologist, so we have the 2 limits to life on either side. So then we always do experiments around what's the temperature of the fluid going to be?
What's going on back there, Sean? What's going on? Okay, so we're having flaky communications, according to Sean. So we're just going to sit here and look at the shimmery flow while we figure it out. So we're in the same hydrothermal vent field that we were yesterday, but yesterday we focused on sampling the high temperature end member, the hottest hydrothermal fluid in this vent field, which is 398 degrees Celsius, and Jeff got a beautiful sample after a lot of positioning and figuring out how to do that. And now we're focusing this dive for a few hours on sampling some of the lower temperature fluids for both chemistry and microbiology. But being able to measure temperature is really important because we use that to guide our sampling. So that's why we're sitting here as we figure out how to get an ICL to work and get a good temperature. I'm going to go get a cup of tea.
So you can see in the back of the control room in the red top is Jeff, who's currently working on getting the sampler ready. In the other view on the right hand side of the control room is Casey, the pilot. Just behind him is Chris, and I'm on the far right talking in the microphone. Behind them you've got Lars from our ship's crew, who's just popped in to see what's going on, along with Lisa.
Just a quick update. We're having some trouble with our data communications up our optical fiber cable. That's true for both Jeff's 2 sets of high temperature vents and for the low temperature sensor that Chip has with him. So at the moment we're just leaving the camera focused on the diffuse flow site. We figured we might as well just record some nice video for you while we're waiting to sort out our technical problems. It's a bit more interesting putting up the test card panel that Leighton downloaded for us earlier in the cruise.
So what's today? What day is it? Tuesday?
Yeah.
So Wednesday, Thursday, Friday are...
Wednesday, Thursday at Von Dam.
Okay.
Friday, Saturday back here.
Okay. So we are storing the IGT. We did not take a sample. Having some trouble with our temperature probes. So we're going to do some troubleshooting on the seafloor. Try to figure things out. We'll give you a better view in just a minute.
What do they have that can try?
So what exactly are we going to do?
We're going to lift up that number 3 paddle,
Okay.
which is part of the connection to the seafloor.
Yep. So we're moving around some of the ICL paddles that allow for communication between the sampler and the vehicle to measure temperature. We're just wondering if they're on a bad path, and so they can't talk to one another. We're basically tidying our basket.
We have nothing at the moment. We can't end up worse off. We've got a little bit.
I thought you took straight back out of it. Oh, we did the bit before.
We had about 30 seconds or so, and 30 seconds is like around to
So the thing Nereus is holding in his hand is the loop part of the inductively coupled loop. Is that what it's called? We're just trying to see if we can get better temperature readings by moving that around.
1427. Oh, while I do that, I'm just going to go grab my salt. Anyone else need anything, Casey?
okay.
Another
Ginger ale.
The ginger ale. The 0 for the hero again. Anyone else? No.
Thanks, Dave.
She's great. I can eat cookies and soda for breakfast.
Casey's eating chocolate chip cookies for breakfast. Looks like Mario's eating some Cheez-Its of some sort. Cheetos. Generic Cheetos. My son loves Cheetos. So Jeff's in the back of the room, seeing if we're getting any better temperature measurements. Talking to the engineers, trying to get things to work here.
Where's Malvin's place? Probably from Jason.
Oh, there's a squat lobster. Right? Or is that anemone?
Jason's late. Better not be out and down here.
What is that?
Center of the
Okay. How many cookies you got down there, Casey?
I have one left.
Oh, I'm not going to take the pilot's last cookie.
You can have it. I've had two already.
No, it's okay.
Alright, we're going to put that back. Pick up the IGT again to RS.
It's not working still?
No.
How about that? Yeah, you might as well.
Doesn't look as vigorous though. No.
No.
That's like
Not as bright white.
Uh-oh. We've stirred up a dust cloud here.
We've just proved that KC is pretty well calibrated. So we've had communication long enough.
anything better?
After a late night, people are starting to make their way out of bed. People who didn't need to be here for the launch.
Put this one down.
Yeah, so we put the. Do you want it back on the boat?
Yeah, you might as well just put it back on the boat.
We'll pick the IGT back up. And we'll just go for it.
We're gonna just go for it, try to get a sample here. Our temperature's a little bit flaky, but we're gonna use a combination of our eyes and the temperature we can get to take a sample.
Well, Casey guessed 121, and that was the hardest temperature we saw, so we know the pilot is very well calibrated with the temperature sensors.
Casey is calibrated.
It's a Casey is calibrated.
Does it take 12 to 14 hours?
Nope. Near seconds.
We don't even let him sleep. Second.
So we're back. Trying to get back in position to the hot chimlet site. It's just to the right. There it is. Every time we move, we stir up this cloud of basically hydrothermal fallout. Those black smokers and other venting, they're loaded in metals, and it falls back to the seafloor. Basically, it looks like the whole seafloor is rusting, which in many cases it is. There's basalt, which are the black shiny rocks. As it gets oxidized, rusts out basically, the iron gets oxidized. Both by microbes and just by water circulating through the rocks.
Remember the days when like you went to sea and all you had with you to sort anything out is whatever you packed. And then today, I'm just impressed. It's like Sean is just emailing somebody, an engineer, to do some troubleshooting on that stuff. Adrian Glover said there's a photograph of what chimney what it looked like 4 months ago.
It has its pros and its cons. Everyone can send you the emails about little things as well.
But still, how you can actually use, rather than just being a distraction for what you're trying to do at sea. Really empower the research a lot better.
You have great support if anything does go wrong these days.
Question on Twitter was if we saw a snail shell just below the arm, and we think we did. Maybe we'll have a chance to zoom in on that again. Right now we're going to get the IGT back out and get a sample.
Same one?
Same one, yes.
IGT-4.
Nereus 001.
It's me. I'm going to call it 004. Some of the key video clips we've been giving numbers to as well. ask about that?
I said, are these going to be sample numbers?
Oh, did you? Sorry, I didn't hear that. They were. Samples 1 to 8.
Okay, so I'll put that in.
I hadn't thought of doing that before, but we did it last night for Brennan Phillips for his videos. So we did that, so Adrian Glover will be number 1.
This 001 whale bone.
Yeah.
Nereus
002 and 003 are both hot chiblets.
All right, we're getting IGT back out of the basket. Going to take a sample here at the hot chimlet area. We're at 4985 meters. We're at the BB vent site, part of the Piccard hydrothermal vent field, located at just south of the Mid-Cayman, Grand Cayman Islands, and the Mid-Cayman Trough. The deepest part of the trough is 6,500 meters. Our survey back in here, 2009. We did not find any signs of venting in those deeper waters, but we did find this site, and our British colleagues as well have visited, and just the last couple days our Japanese colleagues have visited, so the Mid-Cayman Rise is becoming a very popular place. The combination of interesting chemistry and great depth and interesting animals makes it a really cool place to do deep sea science. So we have that IGT out of the basket. I'm going to pan up here in just a minute so you can see it in the manipulator arm. So it's basically, we only have chemists in here this morning, and me. I like to pretend I'm a little bit of a chemist. Microbes are really good chemists. And the ROV gang. Our marine tech, Leighton, he's logging for us. We keep a searchable database where we log all our events with our XY location, our depth, our heading, what exactly we're doing, so that when we go back to the video later, we have a record of what happened. And of course, Chris German, directing the dive. So we're making our approach. Is that the same spot? So these fluids are sort of a mixture of that 398-degree fluid that's been diluted with seawater beneath the seafloor. And we have no idea the time it takes for that mixing to occur, where exactly it occurs beneath the crust. Does it occur in the shallow seafloor, a few meters or hundreds of meters beneath the seafloor? How long does it take? Is it a straight path? Is it a windy path? There's a lot of interesting questions about subsea floor hydrology that we don't know the answer to. And what we mainly do is we collect the fluids coming out of the seafloor and try to make inferences about what's happening beneath the seafloor. So another way to get to some of those scientific questions is if we were to drill and actually get access beneath the seafloor, but it's very hard to drill an active hydrothermal vent, especially one that's not sedimented. It's just hard to set the drill bit. So instead we use these fluids that are naturally emanating from the seafloor as our proxy for understanding what's happening beneath the seafloor. And we do this for chemistry and microbiology in particular. Okay. So we're just switching our camera feed to our zoomed in view. Try to find the best spot for taking the sample. It's funny it showed up there before it showed up there. Good morning. Cindy Vandover has joined us. I had to talk about shrimp, Cindy.
She couldn't resist it. Not because we made her. She just couldn't stop herself.
Someone wanted to know how... Okay. All right, so we have a zoomed in field of view. Help us guide exactly where we're putting that snorkel. Are we getting any temperature, or is it just... So we're getting some temperature, but it's not super reliable. But we think we can still pull it off. So you can see we're putting the snorkel right into the hot fluid. We got Jeff in the back of the room monitoring temperature, and when he's happy with the placement of the sampler, we will take a sample. Looks like we have quite a few people watching out there. Good morning. We launched our vehicle at 6 a.m. from, right? 6 a.m. East Coast time. Got to the bottom around 9 a.m. It's about a 3-hour trip through the water column. Leighton is telling me that in the time it takes us to go 5,000 meters, we could fly from Jamaica to New York. We're at 54 degrees Celsius. We know it's hotter. We got 121 earlier.
Say again, Jeff?
Did Cindy see that shell? What's it at? Adrian is telling us we should pick it up. No, the shell.
We haven't got anything to pick it up with.
Yeah.
I can get that.
No, you couldn't.
We had more time.
Oh, yeah, get more time.
We'll make sure Cindy sees it. Jill McDermott has joined us in the van. She's Jeff Seawald's grad student. She's participated in all of our Mid-Cayman Rise cruises. It's basically the bulk of her PhD is going to be about these vents, their chemistry. If Jill's mom is listening, hi. We're working at a, yep, hi Jill's mom. Jill helped recover the CTD last night. She did a good job. We're locking in right there. We're at 49.85, starting our sample, IGT number 4. What does the temperature say? Okay. Ooh, that's good for me. 85 degrees Celsius. Sorry. You know, got to be a little bit selfish. Correct. Started at 1448. So these IGTs are programmed to fill for 2 minutes. They're sent to the seafloor pressurized with seawater, a little bit of oil, and that volume is displaced by the sample. And then they stay at the pressure the sample is collected in all the way back up through the surface into the lab. And then they carefully extract the gases and then the fluids to do the full chemistry. When you don't collect samples with these types of gas tights, you lose a lot of gas. It comes out of solution as you move up through the water column. So to get the most accurate numbers, precise, I guess is the right word, precise numbers for concentrations of volatiles. 100? Accurate. Sorry. I always make up accurate and precise. I guess I'd make a bad chemist. We need to use these types of samplers. Last night we got a really beautiful 398 degree end member fluid from this site. And now we're getting about a 85 degree fluid, which is a mixture of that hot end member and seawater mixing beneath the seafloor, cooling beneath the seafloor, circulating beneath the seafloor in some manner. There are lots of microbes that can grow at 85 degrees Celsius. Many of them belong to the domain of life archaea, but there's also some bacteria that can grow that high as well. A lot of them eat sulfur and hydrogen, iron. They make methane. Very few can tolerate oxygen. In general, oxygen goes away above about 25 degrees Celsius. So we're especially interested in sort of that boundary of life, the upper temperature limit around 120 degrees C. So we're done with IGT4. We're going to stow it away. And are we going to do another one? Okay. We're going to take another IGT here. Then we're going to switch over to super sampling to get samples for microbial analysis as well. So enjoy the view because we're going to be here for a little while. So how did the temperature probe behave? It's just flaky, but do you trust the number?
Yeah, we can swap it back out now.
When the communications were good, the temperature looked reasonable.
Okay.
Bad communications.
It was close to when you started.
Yeah.
Did Craft Robotics make the arm? Craft Robotics made the arm. It's a 7-function arm, and it weighs 104 pounds. 110 pounds? Less than 110 pounds. I should know. My husband built it. But it had to be designed light and special so that high efficiency so that it could operate at great depth. This vehicle can go all the way to 11,000 meters. It's the world's deepest diving remotely operated vehicle, I believe. I went to the bottom of the Mariana Trench in 2009. So 5,000 meters is pretty easy for this vehicle. And for those of you who don't know about Nereus, it's a one-armed ROV. It can also work as an autonomous underwater vehicle. We remove the basket, which includes the arm. And it can be set free from the ship to run, make a map, has sensors on it, and then it can be converted back over to ROV. So unless all right, we're back. We had to cycle our video. We've got IGT number 6 in the manipulator. We're doing some cable origami. We're not trying to tie knots. We're just trying to clean things up here. Hi, Chip. We're just firing off this IGT and then taking some super samplers. Super samples. My least favorite part about being at sea is having to wake people up. Thanks, Jeremy. Jeremy is our video guru out here. He's part of the Falcor crew. He's responsible for making sure the video is going out to you and archiving it and things like that. So we're positioning IGT, the next IGT. 5? I already forgot. 6? Nereus sample 5, IGT 6. In the same spot. Replication is a good thing. So again, we use both just a visual from the video and temperature to help guide our sampling of these vents. We have Jeff in the back of the room who's monitoring temperature. Jill is standing next to Casey and Chris, helping them find the highest spot, the best spot. We're at 36 degrees Celsius. 41. Yes. We're at 75. 80. 81.
80.
Temperature right there is about 80 degrees. It's nice and steady. Oh, hold it. We are locking in there. Jill wants to move it a little bit. It's her PhD, so the woman's got a say in things. This particular woman. Gone down a couple degrees. There we go. Again, we're using both temperature and visual video to help us get temperature probe right in the flow. If your video is frozen for any reason, just try refreshing your browser. We had to cycle video a few minutes ago. Should be back. So even though it looks like we've only moved the intake about 1 millimeter, we've dropped 70 degrees. So these are very delicate operations. All right, we're locking the arms in. Excuse me.
Stop.
You move a little bit.
Oh, what do you
Can I drop down to like 850?
What?
Wood nails, Bowie nails. Give me trouble. That's it.
That's that.
50, right? 58.
The upper temperature limit for life is 122 degrees Celsius. It's set by an archaea, methanogen,
266.
Yeah, 50, right?
cultured by Ken Takai, who just dove in Shinkai the other day. Saw Ken on TV. But most scientists believe it might be a little bit higher,
Yep. Is that good?
and that if you put the microbes under pressure, that they might be able to grow higher.
That's good right there.
All right.
Okay, steady.
70s. There we go.
All right, we're firing off the IGT at 1503.
Start IGT6, newest sample, 001.
Area sample 005. We're here at 4984.5 meters. Our heading is 108. Hot Chimlet, BB Vents Mound, Piccard Vent Field, Mid-Cayman Rise. I thought earlier we saw 120. Ah, you think that was flaky? Hey, Chip. Hi. All right, we are done with that sample. So now we're going to switch over to my sampling, the microbiology sampling.
Happy to narrate this still?
I'm happy to narrate. I got Chip in the back of the room. We use a slightly different sampler designed by Chip Breyer. It's called a super sampler, suspended underwater particulate rosette. And over the years, we keep messing with it to make it appropriate for different types of sampling. And what?
It's a super sexy sampler I have.
Don't make me say that word.
Ay, caliente.
So in this situation, what we have bottles on the super sampler to collect whole fluids. And with those whole fluids, we do things like try to culture microbes, and we count the microbes, and we do activity experiments. But we also have on the sampler filters so that we can concentrate biomass from the fluids, liters and liters of fluids, without having to bring all that water up. And so the super has both filters on it and those whole water sample bottles. And we can also do chemistry on the whole water bottles. And what I usually do is I just follow Jeff around. Wherever he takes a high quality chemistry sample, I try to take a microbiology sample so that I have good data about concentrations of things like hydrogen and sulfide and iron and other things that microbes like to eat. So we also have a temperature sensor at the intake of the super sampler. It's been a little bit compromised, but we'll try to use it. We basically want to sample the exact same spot. Hot chimlet. Super sampler, 4985 meters. Are you serious?
It's serious.
Well, we'll find out how real that temperature was then.
I think it's real. I think
We have a sense for when the temperature is real.
Basically the same number from each of these other struggling.
it's just about to put 3 back on.
It's on. Now we want the super sample.
So those loops you're looking at are part of the inductively coupled link that allows the sensor at the tip of the samplers to talk back to the vehicle and measure temperature. We have 3 loops because we have 3 temperature sensors down there. We have 2 at the intake of the IGTs and 1 at the intake of the super sampler. So now Casey is grabbing the intake of the super sampler. You can see we use sophisticated tools like rubber bands, bungee cords, zip ties to keep things on the vehicle. Walking into the Nereus tool van is really awesome. There's lots of every fitting and bungee and zip tie and screw you can imagine. And it's really well organized, which I imagine was Casey's doing. I wish my house was that organized. I wish I was that organized. So all of our samplers have T-handles on them, which fit snugly in our craft arm. It's important to get a good grip so you don't drop it. Each what? Each arm? They're pricey. They're custom made by Jeff Seawald and his Hui machine shop.
Our English pounds. That's how much they cost. In Casey's world, payload is the most expensive commodity. Worth its weight in water it is.
So we're getting the super sampler out. Gonna put it back in a hot chimlet. You can see we have a cables and tubes and there's a rubber band that just flew off. All right, we put the super sampler down for a minute because we knocked out over the ICL loop that we need to measure temperature.
It's going to be an exercise in frustration if you try and keep that up.
Okay.
Plus it stopped working just before it fell off.
Okay.
Well, I didn't leave, it flipped back to difference. I think you're going to drive yourself to it. It's just going to pop off and just tell it to keep it on.
So there's going to be no temperature? Or flaky temperature?
Well, it won't be any if you don't put it on, but I don't think it'll be connected to the hose. Every time you pulse the hose.
Okay. All right, well, we know what the T-max is here, so we should probably just proceed with the sampling. It's okay.
As you said, you flew blind last year.
Yep.
Expect to get 2 samples without temperature, then one that may or may not.
Yep.
Or we can always try it again at the other place. If this one we already know the answer.
Right. So we're grabbing the intake of the super sampler again. Then we'll zoom back out to show you the pretty vent that we're sampling. This site is called Hot Chimlet. It's located at 4985 meters. It's one of the deeper sites here at the world's deepest hydrothermal vents. Yep. So that area of diffuse venting in front of us, you can see the white shimmery flow. There's shrimp around it. We're going to zoom into that, and that's where we're going to take our sample. Chip, do you have written down which ports are what?
Yes.
Okay.
Port 1 is full 1.
Okay.
Go to the same part of it. Should we zoom in and have a look at it?
Yeah, let's zoom in, and we're basically going to try to do the exact same place that Jeff sampled. We're switching to the zoomed in view. Both views that you guys are getting back home are courtesy of James Cameron's lights and cameras that went down on the Deep Sea Challenger to the Mariana Trench last year. They've been donated to HUI. So we immediately took them off the vehicle and put them onto our vehicle, Nereus. Thanks, Jill.
That always gets people out of bed.
All right, we're getting the intake into the flow. So, Jeff, we have nothing coming back by temperature, right? Okay. Can we zoom that in anymore, or is that it? Okay. Yep. You're purging now? No. Okay. What does everybody think of that? Sampling by community vote. You think it's reasonable? A little bit north, I am hearing. Yeah, a little bit. Little bit up and down. All right, hold it there. Yeah. Yep. All right, we're going to purge the pump before we start the sample. We've got the arm locked in. We are purging. Whole water bottle, port number 1, once we're done purging the pump. Don't start that sample yet, Chip.
No, I'm just purging.
Okay.
Keep purging because Casey's arm is twitched, so we're going to make sure we're in the right place. And
Sometimes the arm can be a little bit twitchy.
purging fog again now. Is that getting stable?
Well, I won't be able to predict when I'm putting it,
No.
so everything else is stable.
Okay.
The arm has a nervous twitch when it takes microbiology samples.
We're taking it off caffeine.
I'm just exhausted from holding up jump samples.
Casey says that the arm is tired from holding up those big titanium samplers. All right, Chip, it's cleared out, so when you're ready to switch to port 1, let us know. Port 1? All right, we're starting a whole water bottle sample, port number 1.
One whole water and 2 Steravax here, or the other way
I think the other way around. So, Chip, how long are we going to let the bottles fill?
I have a flow count that I have a target flow count.
Okay.
So I have a specific number. I need
To
to flush.
Okay.
2 whole waters and 1 Steravax, or the other way around?
Yeah, it's going to be, Steravax will be in the middle the fastest way. That's the way
The valve goes. Okay.
I'm going to go talk to James about navigation this afternoon.
Okay.
Well,
Ooh.
I'll just do that.
All right, we're having a little bit of a twitchy arm.
Call me when you're ready to move on to the second side.
Oh. So we're sitting here at the hot chimlet site, sampling this 80-degree fluid for microbiology. Right now we're filling a bottle on the super sampler. We took a couple of chemistry samples here. Now we're taking some microbiology samples. This is the hot chimlet site we found in January 2012. Apparently I named it, but I don't remember that. You can see there's shrimp around. And there's some nice hot fluid coming in off the seafloor. The intake of our sampler is blurry because of the diffuse flow. And that fluid's rich in hydrogen and hydrogen sulfide, carbon dioxide, iron. Probably has a pH around 6, 5 or 6. And it's a result of that 398-degree fluid we sampled yesterday mixing with seawater beneath the seafloor. And a lot of different chemical reactions can happen during that mixing, and then it makes its way out where we sample it. So Chip Breyer's in the back of the room, monitoring the sampler, and Casey's holding the arm in place. And we're pumping at a rate of about 250 mils per minute. And the bottle is a liter. So we need to pump for a little while to flush the bottle of the Millicue water, the clean water we sent it down with, and replace it with vent fluid. We are stopping that sample at 1527. Now we're going to switch to a filter. So that first sample collected whole water that we use for cell counts, for culturing, for activity measurements, for chemistry. Now we're going to switch to a filter which concentrates the biomass. Has a very, very tiny pore size on it. Microbes are really small. They're about 1 micrometer, which is 0.001 millimeters. Right? Yeah, they're really small. You need a high-powered microscope to see them. But what the filter does is it captures all the microbial biomass on that surface so that we can sample it. And this is Nereus sample number 7. Port 2, it's a Sterivex filter. You can just put filter. Let us know when you start, Chip. And we're going to try to pump a few liters through that filter. And when we get it back in the lab, we can extract the DNA or the RNA, the nucleic acids, the code of life. All right, we're starting that sample at 1528. A lot of the microbes in these environments are really, really hard to culture, and so we use molecular biology. We can look at their genetic code and infer what they're capable of doing and how they're surviving. That one over there. The one on the top, I think. Oh, yeah, I can't read that far away. Oh, great. So, yeah, are we going with the Doppler XY on the... Is that what we should write down from the nav screen? Or the cursor?
The cursor, the Doppler thinks we're like a mile away right now.
Okay.
The cursor position is where, when we saw this, we dropped a marker based on the nav at that point. So I would go with the cursor position, which is X of 3365, Y of 5226.
Great. So when we record our samples, we record our XY, which is basically our latitude and longitude on the seafloor, the best navigation we can get. We use a lot of different navigation tools on the seafloor. We can't use GPS, but we have a pretty good map, and we use something called ultra-short baseline navigation. We basically have a pole that's sticks out the bottom of the Falcor. And then we have a transponder on Nereus, and we try to get them to talk to each other so that we can figure out where Nereus is in relationship to the ship, and the ship is using GPS to figure out where it is. But that can be challenging. Ultra-short baseline doesn't work that great at 5,000 meters, where we're operating today. And also Nereus can wander pretty far away from the ship. It has that light fiber optic cable that's sending back our telemetry and our video, lets us control the vehicle, but we have to make our way over large sulfide mounds. There's a lot of topography here. Temperature can mess with that as well. So there's a lot of challenges to doing navigation on the seafloor. But today we landed right where we thought we were going to land. So Nereus guys did some work last night to make sure they understood what was going on with the navigation, and we basically landed right where we wanted to be today. So kudos to them. So the reason we're just sitting here is that we are pumping liters and liters of this fluid through a very small pore size of a filter to collect microbial biomass. And our pump is pumping at about, I don't know, between about 100 and 200 milliliters per minute. It starts to clog after a little while as we get more biomass and we get some particles from the minerals and sediments around us. And we, these fluids at 85 degrees C have about 10,000 cells per milliliter. So we want to collect as many cells as possible to get a sense of who's living there, what they're capable of doing, how they're related to one another, and what their activity is like. So I think I've seen everybody in the science party pop in and out now this morning. It's almost lunchtime. Lunch is the big meal around here. It's a big deal. It's really good. It's super fancy. And most of us didn't have breakfast, so lunch will be extra special today. Casey's been eating Chips Ahoy and cheese puffs for breakfast. Chocolate chip cookies and cheese puffs. They really are generic cheese puffs. Jamaican cheese puffs. Even better. Our ship left port from Montego Bay when we had our switch over from AUV to ROV. And it was hot there. It's hot here. It's about 100% humidity. But we're nice and warm, nice and cool, excuse me. Air-conditioned ship on the inside. Oh, we got a twitchy arm. Just try to poke it back. We'll keep the pump running. I will. We're still in the flow. Sometimes the arm has a little bit of a twitch, so Casey has to push it back into the flow. That looks good. So when you take these long filtered samples, you're basically getting an integration over time of the fluid. And just a snapshot of what's happening in this 20 or 30 minute period. So there's a snail shell. Actually, some people on Twitter were asking what it was. And I said, I don't know. And they said, ask Cindy. And Cindy wasn't awake, but she's awake now. Let's see, Adrian Glover said, Buchanid Isifo? Probably Cindy knows.
We gotta do our interview so I can finish editing my thing.
Okay.
I have a 10-minute Nereus documentary pretty much all edited up.
You want me?
It needs like an introduction and a...
Hello, I'm late. I'm going to introduce you to some very special people in a moment. But first, take them at face value. How much longer on this one?
You said you were about to see, or Robinson? We are about halfway there.
Okay. Well, in the meantime, show everyone in the van so we can introduce ourselves.
Hey, Jeff and Chip, you're on camera. Can you wave? Otherwise...
Yes, special.
You did it right, Chip. There it is. That was awesome.
That can go with the outtakes.
You're right. All right, Mario, can you wave? There's Mario, Nereus engineer. Casey, our pilot. All right, Jill. I'm down at this end. Got Leighton next to me. That's... Oh, James just came in. The camera's on. Turn around, James. Say hi. Oh. That was funny. It does. It looks really awesome. So last fall, our team had the opportunity to visit NASA's Jet Propulsion Laboratory, where one of our scientists, Max Coleman, is, and we got to see the space flight control room where all the missions are landed from. It was really awesome. To be honest, though, they had like this many screens. They were just bigger. Like every screen was maybe 4 times as big. Yeah. Yeah, it's like an amphitheater, and then it has a viewing room above it. And that's where, so the Mars rover that just landed, that's where they land it from, and then they go into their secret spots around the campus. But we also got to go and see the Mars Curiosity rover. They basically have a duplicate of it in Pasadena, California, and before they run any controls on Mars, they do it on in the Mars yard in Pasadena. They have like big boulders and other, obstacles that the vehicle might encounter up on Mars. It was really cool. Yeah. And we met one of the scientists, and he was very tired. He was just coming off shift, and they've been keeping Mars time. So a Mars day, I think, is 30 minutes longer than a U.S. Earth day. Not a U.S. day, an Earth day. So they're on they were on really weird time, and they were slowly going to switch over back to Earth time. But it was really neat.
Imagine saying you're on Martian time for your face.
I know.
I'm on Martian.
I'm on Martian time. That's too late for me.
It's longer?
It's a little bit longer. No, I think it's only like a half an hour longer or something like that.
Catches up with you eventually.
Yeah. Yeah.
So you're going to get more and more
Off, exactly.
The way I
Those of us out here can kind of relate because the days just kind of run into each other, and I don't know, time doesn't really matter.
It matters.
More time we're
Is
doing.
today Tuesday?
Today is Tuesday. It's almost lunchtime.
80 more days out here?
We have 5 more days on station. 6.
So what is that, 6 and wake up?
Julian days earlier were on 176 at the moment for people who do Julian days.
I don't think anybody out there does Julian days.
12 to wake. 12? 2 times per day. 2 times per day.
So we're in 4, wait, is there 6
So
more
the-
days out here? 6 and a wake up, right?
Well, I don't know. You wake up and go to bed a lot out here, right?
Well, you say however many days you have, and you subtract 1 and say wake up. So all you have to do is wake up that day.
Yeah, but if you're Chip, you go to bed 4 times in a 24-hour period for like 2 hours each time. So another cool thing about the Mars rover that I learned is there's a satellite going around Mars, and so they can send commands directly to the rover, but it's quicker if they send it to the satellite when it's right over where the rover is. So they tried to coordinate their big computer updates and all those things for when the satellite's aligned with the rover. It's really neat. So they're doing all that by radio.
How often does the satellite pass?
It's pretty fast. But, they launched the vehicle, 13 months before it actually arrived. And so the first thing they had to do was update all the software because things had changed. Yeah, everything. They had to, that's exactly what they had to do. So that was a lot of data to send up there. So they did it via the satellite, and it just goes better. There's about a 10-minute delay. But we can't use radio at the bottom of the ocean. Radio doesn't travel through water very well. So in some ways, it's easier to talk to a vehicle on Mars than it is at the bottom of the seafloor. So this project was originally funded by NASA, which is why our team was at NASA for our final team meeting, because we're interested in robotic exploration of other planets and moons, and also the origin and evolution of life on our planet and possibly other planets and moons. So thanks, NASA. Can you pause that, Chip? Let's just stop it. Yep. We just stopped that sample at 1542. So how long was that?
We started at 15:28, but you got a lot of water.
Okay. So Casey, we want to put it back in, and we're going to get one more bottle, which should take less time. Did the pump slow down at all?
Slow down a little bit. Yeah.
Okay.
I think on average we're pumping, it's not tailing off as fast as last year because the tubing runs are longer.
Uh-huh.
Delivering more constant pressure.
Okay. That's good.
Getting your sample a little bit quicker.
Great. So we're going to pop our sampler in there one more time to get another whole water sample. Then we need to go get our fearless leader to take us to another diffuse sampling site.
Okay, this will be 8, 4, 3. Is there any filters?
We doing port 3, Chip? Okay, port 3, it's a whole water bottle sample. Nereus sample number 8. Yep, whole water. Yep, that looks good. Chip, whenever you're ready. Starting bottle 3 at 1544. We're at 4984.5 meters. We're heading 108. We're at the hot chimlet site. This is a liter of water, which means I get to stay up very late tonight, inoculating a bunch of anaerobic serum tubes, filtering water, fixing water in various ways. Last night the lab smelled really bad because those fluids we collected were loaded in hydrogen sulfide. But those of us in that room really like those. Like that smell. It's the smell of life. Smell of the origin of life. It smells like rotten eggs to most people, but to us it smells like life. Well, if you include the CTD, which, what, 240 liters? No, it's more than that. 24 times 19, right? Yeah, so 470.
You ever wonder that in years from now the ocean won't have any water?
I don't think we're going to sample all the water in the world's oceans.
I wish I had a Twitter account.
Yeah?
Because I would sit here and
You would wonder about those things? No, I don't.
Dan had to compare to Jason. We had to adjust our program so that we had delay. We didn't know it. Longer, bigger window. We had been waiting for lunch.
So what's up, Casey? You going to let anyone else drive the vehicle? Okay.
Well, the way we structured it is that I do, I'm supposed to be standing a 6-hour watch, but the dive's only going to go till 2, then it
Yep.
doesn't make sense to waste 15 minutes to change it.
So we're going to be taking the sample for a couple minutes, then we're going to make a move. Julie, do you mind grabbing Chris from in there? Tell him we're almost done sampling. Yeah? That's always scary. And then where are we going to go?
Well, I need to get you and Jeff in a huddle here. Hey, Jeff. Have one say for you and Julie, please.
Chris is rejoining us. Did you stop the sampler? No. No, no. Sorry. That, keep going. You tell us when you're done. We're just switching the video feed. We're just in this area of venting with sulfide rubble around us. Some shrimp, some nice hot fluids coming out. Sparse shrimp. The rest of this dive is going to be Reconnaissance. In 2009, we left a titanium sampler on the seafloor, and we are going to go search for it. Search and recover mission. It's more accurate description. So it's been on the seafloor since the fall of 2009. We have a pretty good idea of where it is, but we're going to go look for it. It belongs to Jeff Seawald. We went down a little bit light in our basket so that we could recover it if we find it. So over to the... All right, we're stopping that sample at something 1551. And we can stow the super sampler. Correct. Good set of samples. Thank you, Casey. We're going to do a little drive-by to check out some other diffuse venting, and then we're going to make our way over to where we believe the missing sampler is. All right, we have stowed the super sampler. Going to do a little bit of a drive-by, some other diffuse venting. Hopefully we can get some pretty shots for you as we make our way out of this hydrothermal field. We left our sampler a little bit away from where all the venting is. So we have James Kinsey in here. It's our knows the seafloor really well in this area. He is he's helping us find the targets for where we want to do the rest of our sampling. I mean, not the rest, we might do the rest of our sampling on future dives. We're not going to do it on this dive. All right, we're going to heading on a course 160, 180, basically due south. So one thing we have to be really careful about with Nereus is it has a very small fiber optic cable that connects it to a depressor weight, which is at about 1,500 meters. Then that depressor weight is connected to the ship by a more sturdy steel cable. So we proceeding on heading 200, just west of south. And we always have to watch our path because we don't want to cross over our own cable too many times. We also don't want to get our cable wrapped around anything or melt it. So it's very important for... Oh, look at that big anemone. Can we, is that camera panned out or is it, oh, seems like it's a weird angle. That is a big anemone there. Right. Oh yeah, look at that. That was in that. See all those shrimp there. That was in that other picture Adrian sent us, facing the wall. Right now we're bumping the wall a little bit. Okay. Just old. Oh yeah, there's the furry wall covered in microbial mat. That white filamentous microbial mat that we saw quite a bit of yesterday. These microbes are firmly attached to that rock. Last year when we sampled it, we couldn't manage to vacuum the mat off, so we brought a rock up and scraped off the filaments with a razor. Oh, and look at the shrimp swimming away. Probably scared by the thrusters, the vertical thrusters on the vehicle. So we're coming up in the water column a little bit. All those white patches on the seafloor are swarms of shrimp and microbial mat. This is where we're going to have a target for sampling 2 days from now, 3 days from now, we hope. We'll be back here. You can see the shrimp swarming. Yep, that's all shimmering water in those areas where you see the shrimp congregating. The chemosynthetic bacteria that are their source of carbon, their source of livelihood, need to be right on the interface between the hot sulfide coming out of the vent fluids and the cold oxygen in the seawater and the fluids around them. Shrimp galore. Last night we slurped up a bunch of those shrimp and yep. So we're just trying to get some good video of this spot so when we come back next time we can take some more samples. We've filled up our IGTs and our temperature probe is compromised, so we are going to move on to the search and recover part of our mission, which is looking for a water bottle we placed, not happily, but placed on the seafloor about 4 years ago. So it's getting loud in the van. Everybody's up because it's lunchtime. And we're going to shortly be making our way up and over this cliff face with all these little pockets of venting and shrimp. What did we call this site last year? This isn't the crack site? Oh. Oh, okay. Okay. Can see some anemones in there. So look at all those shrimp up there in the upper left-hand corner. Every time you see large congregations of shrimp, you know there's diffuse venting coming out. Can be challenging to sample. Ooh, there's a nice big anemone. We also see some orange staining, white staining on the rocks. It's basically rust. Some iron rusting of those rocks. Are you talking about that black one? Oh, yep, I see it. Yep, see it down there. So as we make this little climb up the wall. Down, right there, right there. We're dropping navigation targets so we can find our way back here on another dive. We have limited bottom time today. We have to leave the bottom at 2 p.m. Eastern Standard Time. So we're trying to make good use of our time. We came down a little bit light so that if we do find our missing bottle, we can put it on the vehicle and come up. Nereus has a payload of about 110 pounds, which is not even the weight of the average grown-up. And so we carefully weigh everything in water so that we have precise measurements so we don't get too heavy. There's some pretty white, furry microbial mat on that rock. Those are sulfide oxidizing gamma proteobacteria. It's a class of bacteria. Gamma proteobacteria live just about every environment you imagine, from the human gut to soils to deep sea hydrothermal vents. A very successful group of organisms. You've probably heard of E. coli. There's lots of good E. coli. Oh, there's a focused area of flow there. Down and to the left. We're just passing over it now. So you have good E. coli in your gut that can get bad E. coli. You've probably heard about the scares from contaminated food. But most of the gamma proteobacteria in the world are nice. They're not out to harm you. They're just out making a living, such as these sulfur oxidizing bugs at the bottom of the ocean. And these types of white microbial mats are seen in a lot of sulfidic rich environments, like Yellowstone hot springs, Iceland hot springs, cold seeps, and lots of places. And we're interested in how they get there. How do they know to stay? How do they talk to each other to form these amazing matte surfaces? Microbes can actually communicate with one another through a process called quorum sensing. They basically release molecules, and when there's a quorum, enough of them, with enough molecules in the water, they can act together as a community, which is pretty remarkable considering these are some of the oldest organisms on our planet. It's believed that life originated around 3.8 billion years ago. So microbes have had a lot of time to diversify, pick up new tricks, and to colonize almost every habitat on the planet. Okay. So your little lesson in microbiology is over for the day. I'm going to hand the microphone over to Chris German.
Yeah, thanks, Julie. Okay, so it is just gone 12 o'clock on the seafloor. We have 2 hours to play with. And thank you for that climb through the diffuse flow area, Casey. I think we're now ready to go head off to search for Jeff's missing bottle. We're at a depth of 4971. Mario has a target for you in the bearing is 278, 280, and the range is 450 meters. So okay. I think you guys are off the... Absolutely, we've just got to drive 400 miles... No, not 400 miles. 400 meters through the water column. It's just going to feel like 400 miles if you're watching at home. And, yeah, we have to basically head out across a gully, and then we hit the next wall, and that's where the other super sampler should be. So... Sorry, IGT, not super sampler. Yeah, we weren't far away. So is the Doppler reset done? Thank you.
What is our job?
we're going to get deeper. We're going to be at about 4990.
Is there anything shallower?
No, it's basically downhill, so I just brought you up shallow enough. If you want to come up to 4950 maybe, I don't know how far you want to go. Basically, it's we're going to go out over a gully and back up the other side. James is creating the underlay for you, that's why you can't see it. But what we're going to do is I'm going to log every few minutes what our depth and heading, or depth and altitude. And if you have any way you can gauge what you think forward velocity is.
We can tell you what the vehicle thinks it's for.
Okay.
Speed over ground right over here.
Okay, so let's do that every 5 minutes and we'll start in 60 seconds.
Chris, did you eat already? No. Do you mind releasing me and I'll go eat?
You want to be back to pick up that bottle, don't you, Casey? It's a matter of personal honor with our expedition leader, Casey Machado, that he was the person that, was it you that actually lifted the bottle out and put it down on the seafloor? So Casey is determined that he wants to get Jeff's bottle back in person. So he's organizing when Chris Taylor takes over for him as the additional pilot for the day to make sure that Casey's back in the saddle when the moment comes. Hoping that it does. we're all good now. So, as you can see, we're just changing the screen of what we're showing you at the moment. We are flying through the mid-water column, and the reason we're doing that is we've been doing our sampling for the first half of today's dive, but the second half, from now until 2 o'clock when we have to leave the seafloor, is a search and rescue mission for one of Jeff's titanium fluid samplers that was left behind here on the seafloor in 2009. So we have good coordinates for where it was. We have video we've been replaying of the moment we had to put it down. We were in a situation where we'd got ourselves unexpectedly heavy on the submarine. So we had a choice of either we left Jeff's fluid sampler behind, which is the heaviest piece of equipment we had, or we might have ended up just leaving the whole submarine on the seafloor. So it was, unfortunately for Jeff, it was an easy decision even for him to be able to help us make. That was the necessary sacrifice, but we did promise him we'd come back, and so today's the glorious day when we go look for his sampler that we lost, or left behind all those 4 years ago. So on that map now you can see on the east is the hydrothermal field we've been sampling today and yesterday, and where we're headed due west from here, we're going across this valley, flying out in the water column when the seafloor rises up to meet us at the same depth pretty much, or maybe 5 or 10 meters shallower. That's when we're going to be arriving at the area where we left Jeff's bottle. So we're hoping we have enough time to get that before 2 o'clock this afternoon, which is when the captain requires us to leave the seafloor to get back onto the surface in time to allow for any eventualities while we recover Nereus inboard to make sure everything is all done well before sunset. So the time is now come up for 12:20 here on the RV Falcor. Just to let you know what we've been doing, we have a short dive today. We arrived on the seafloor at 9:30 and we have to leave the bottom again at 2, just to fit in with when the sun goes down in this part of the world. So we are currently hightailing it across the seafloor from one mountaintop to the next. We're flying across a valley in the dark, but you can see the track we're following in our nearest plan. We're basically on our way to try and locate a sampling system that we left behind on the seabed, and the way to orientate ourselves, most of the seafloor we're headed towards is covered under sediment, but there's a large cliff face. You can see that little target 18, that says rock wall target. Nereus is heading that way. We have 140 meters still to go, part of a 450 meter transit, and we're 2/3 of the way there. When we get there, we're expecting to see a cliff full of basalt, and what we will try and do is from there we should have a range and bearing to where Jeff's fluid sampler should be. So that's where we'll be heading next. So we're now arriving at the very cliff wall we wanted to find.
So now, let's do some, so we have a depth.
Are we at the right depth to be? This is the very same cliff wall.
So we were expecting to see that at 4971 or 4951. We're 20 meters deeper. And that's not to
10.
say this is
We're
a
at
big
4961. It's only 10 meters deeper.
Right, and that's not to say this is a big feature that we approached. Remember, we approached it from that
And we climbed up it
so we climbed up
30 meters. It may not be the only one. This may be, you know, if it's an entire pillow mound.
Yeah.
But this is definitely steep without the sediment drape over it. What you're looking at, if you're not familiar with these things, is... do you need some more light on, Chris? They probably had the lights turned off for the transit. Okay. What we're actually looking at here are a hillside full of pillow basalts. Each of these lobes is about 1 meter across. And they're called pillows. Basically, one stacks on top of another. They're about the size and shape of a pillow. This is actually one of the main ways that volcanic lavas flow out on the slow-spreading ridges. The lava actually erupts at about 1100 degrees Celsius on the seafloor. And in the process of erupting, these things chill around the outside. So the outer skin cools and turns solid. The inside remains soft initially and molten, rather like a hard-boiled sweet, except that it's a hard-boiled sweet under pressure, that as more lava flows in from one end of the tube, these things cool and then burst open one after another. So here on a steep slope, you can see that these things sort of crack open. You can actually see radial structures inside them because they're concentric. They're basically, and we can see end-on inside some of these with the fractures we're looking at. Okay, so this cliff wall now, we know in other parts of the seafloor in this area can be tens of meters high. We spent a lot of time this time last year with a colleague, Tom Klasnovica, collecting three-dimensional imagery from a wall like this. So we're heading up, we're starting at 4960 meters, heading upslope, and we'll see what we see. So we're climbing up over these pillow basalts. You can see radial fractures, basically these tubes of lava flowing downhill towards us. And every now and then they've broken open when they were being erupted. Would have been a fun place to be the day all this stuff was forming. And plenty hot too. These things, they cool instantaneously on the outside, but just because they're solidified doesn't actually really make them that cold. They actually solidify at about just over 1,000 degrees Celsius. So just because you can hit them with a hammer and they'd feel hard doesn't mean that they're cold. And that's really the basis of where all hydrothermal circulation comes from, that the seafloor lavas erupt. The fresh ocean crust is put in place, it becomes solid and brittle, but it's still extremely hot, 1,000 degrees Celsius, so that as it then cools and contracts and fractures, that's where the seawater can percolate in and get heated up to hundreds of degrees Celsius, and that's basically how the hydrothermal systems we were diving on yesterday and this morning work. Just to recap what we were doing this morning, we were sampling low temperature fluids, both for Jill McDermott's thesis work with Jeff Seaward and me at Woods Hole, and also for Julie Huber and her group's work for microbiological studies in the same kind of fluids, right around the limits for temperatures that were known to be hospitable for life as we know it. Now we've moved across the valley and we've come to a separate volcanic ridge, which we do not believe to be hydrothermally active, but this was somewhere when we were in our first earlier stages of exploration in 2009. We knew from our chemical signals about halfway through the cruise where the plume signals were coming from, and so we started checking out all the different lumps of about the same size and depth. We're actually at exactly the same depth here as we were yesterday at the Black Smoker chimneys, just about 500 meters away. So the time now is 12:30, if you're still persevering with us. We are still working our way around the side of a new hillside. It's probably hard to see on your camera screens, but there's a wall of basalt right ahead of us. We're just heading towards this cliff wall. We're just making sure we're in the right orientation. Then we're going to climb up the hill. Our range is 30 meters to where we're going to start looking for Jeff's fluid sampler. So we're just going to make sure that we swing around and take a left turn now. So we're actually going to be on the same heading we were on in 2009. And what we know we were doing in 2009, we were heading to the south and southwest. We approached, at this depth, we approached a wall of basalts in this location, which is where we're headed about 10-20 meters ahead of us. And then once we got to the top of that wall, it was soon after that was where we realized we were having such difficulty climbing we needed to lose some weight on the vehicle to make sure we could safely get all the way back to the ocean surface. So we're about to try and reconstruct that path, head in the same direction in the hope that we can actually find Jeff's sampler right where we left it. So the time now is 12:40. We were on a heading of 227 degrees at a depth of 4950 facing into. See, this is more tailless. This isn't exactly the same as what we saw before. So we're now climbing up a wall of basalt fragments, similar to what we were doing in 2009 when we were here before. And we're heading in the right direction for basically at the top of a cliff like this on this orientation, which is where 25 meters shallower is where we ended up leaving Jeff Sampler in 2009. So we're pretty much right in the right place. Little shrimp flying through the picture just here that we're looking at. It's a non-chemosynthetic shrimp. There's still plenty of other kinds of animals that live around here. And we're heading up over our rock wall. Yeah, we actually...
This is just a guidance point in the idea is we're going to vector in, kind of our best recreation of this. So if anything, we're a little bit...
So the good news is that we're flying over exactly the same kind of pillow tubes that we flew over when we were here in 2009. The not so good news is this is pretty much what the entire seafloor of the Cayman Rise looks like as well. So although it's good that it's not something completely different from what we were expecting, what's more important is whether our navigation is dialed in. We actually have some encouragement from that today, that when we went to reoccupy our hot chimlet site today, after some corrections we made to our navigation between 2012 and 2013 overnight, we landed right where we wanted to be. But the issue today is whether or not our navigation is also the same as 2009's navigation set. So we will find out. So the time is 12:50. We're climbing up the hill face, getting close, we hope, to where we think we left, probably within about 50 meters of where we left Jeff's fluid sampler in 2009. So we're keeping an eye out both for the large silver sampler, like the two we have in our basket, but also for a blue scoop net, which is something else that we had to basically jettison anything we could jettison from the basket that day to make sure we got the submarine back safely to the ship. This is by and large the right kind of depth, yeah? Within error.
48, 94.
Yeah, so we're going shallow, so you want a depth of 49.
If we sum these two, we're at 4,900.
Well, that's the value. Should we continue on to the navigation spot, or should
So as we continue this search, we realised that the depth we were at didn't fit well with the terrain, or certainly what we were looking at in the screen didn't look with what the location where we left the bottle. So we're coming back down to the correct depth. We're going to migrate further south along this wall, keeping it in sight. And if we get to the right place, we'll have a clue about that because the terrain will flatten out in front of us without us changing depth. And then when we're at 49-15 meters and we're on a flat sedimented surface above the top of the basalts, then we'll be in good shape. Okay, so for those of you who want to follow along with the continuing quest for Jeff's bottle, we're about to reset our website broadcasts, so you'll need to refresh your browser in about 2 minutes' time. Normal service will continue as soon as possible.